Hi, and welcome back. So this is the lecture content for week 3 for Geology 309, Bringing Fossils to Life. This week we're covering quite a lot of ground. We're going to be looking at microevolution, macroevolution, extinction, and also looking at functional morphology. So let's jump right into it. So let's start out with a light discussion on evolution. Before we get into micro or macro evolution, let's talk about what evolution is and what it isn't. First and foremost, is evolution a theory or a fact? Well, if we look at the role of natural selection, a mechanism that permits evolution, natural selection is defined by Darwin, who coined the idea, as organisms that inherit favorable variation for their immediate environment will tend to survive more often than others. Now notice that this is not a guarantee. It just says that they tend to survive more often than others. And note also that the word immediate is important here. That means an advantage at one time or place may not be advantageous at another time and place. So a classic example of this would be the white coloration on a polar bear. It works very good for the Arctic, doesn't work very good for the jungle, right? So there is no necessary permanent perfect organism. It's just about being a good fit at the right place and at the right time. Microevolution is a mode of evolution in which we're seeing the transformation at the species or even subspecies level. This may or may not lead to the arrival of a new species. And this is factual. This is well documented. This is the reason you get a different flu shot every year. This is the reason why pesticides have to be updated regularly, because organisms become resistant to it. Microevolution is something that we absolutely can observe in a laboratory setting and in various types, like I mentioned, with pesticides and antiviral and antibacterial strains. This is the same reason why if you go to the doctor and they give you an antibiotic, the next time you go to the doctor with an infection, they may not give you the same antibiotic because they don't want you to develop a resistance to it. That is all microevolution. Variation that we talk about here can arise by chance. It's a mutation, okay? But selection is a very powerful driving force. So whether it's sexual selection or natural selection or even just specific breeding like we do with our dogs and cats and horses and plants and so on, all of these are the types of variation that we're referring to. Now let's talk a bit about the difference between genotype and phenotype. Okay. Genotype is your genetic blueprint. The phenotype is the expression. So one way of thinking of this would be that the genotype is the recipe for the phenotype. And to give an example of the relationship between these, let's look at the peppered moth. So you can have multiple morphotypes with one genotype. So the peppered moth is a species of moth that lives in Europe, specifically around Great Britain. Okay, and there are two different morphotypes of this genotype. There is the typica and there is the melanic. The typica is a lighter color with some black spots, and the melanic is mostly dark, black and gray. Okay, both of them have pretty much the exact same genotype. The only difference is one minor genetic difference in what color is going to be expressed. So actually it's the same genetic code, but in this case, one color is being expressed over the other. Okay, and this is a classic example of microevolution because we're not creating a new species here. These are fully capable of interbreeding. Typica and melanic pepper moths can breed and produce offspring. It's what we call a sympatric population, which means that we have these two different forms, but they're living in the same environment. So why is this such a classic example? So the peppered moth, Biston betularia, lives in England. We have these two different types, the black moth, the black one and the white mottled one, or the typica and the melanic. Prior to 1600, prior to the British Industrial Revolution, most peppered moths were the white form, the typica form, and the melanic form, the black form, was only about 1% of the population. After 1600, when the widespread industrial revolution was taking place and there was lots of pollution, smoke, and soot, on the sides of buildings. The black variety made up 90% of the population of these moths, while the white form was only 10%. So they about flipped. Today, it really depends on where you're at. If you were to go to in the industrial neighborhoods of London, you'd notice that the black and white forms both make up about 50%, and that's because pollution is somewhat more controlled. However, when you move away from an industrialized area, we go back to the prior to 1600 relationship, or closer to it. We have 90% being the white form, 10% being the black form. Now, why would this be? Why did we see such a switch? Well, the environmental conditions changed after 1600. We now had new colors on the sides of the buildings with all of that pollution and soot. This allowed the black variety to blend in much better and thus weren't picked off by birds and other predators. During other conditions, when we didn't have a bunch of soot on the sides of buildings, the black ones would have been noticed more easily and the white ones would have been able to blend in more. So this is an example of how the expression of that phenotype, or of that genotype, I should say, is highly dependent on environmental conditions, and that can drive characteristics within the population. Okay, so we didn't create a new species this way, but what we did was we changed what is the average characteristic expressed of a particular species, and that is microevolution. Macroevolution, on the other hand, happens with higher taxonomic categories, so above the species level. This is where we're definitely getting a branch and new types of species and new organisms that are distinctly different from their ancestors. So how does macroevolution occur? Successful macroevolution is not something we can directly observe. This is something that creationists commonly cite. However, we have a fossil record which provides considerably ample, both extraordinary and very, very detailed evidence that it occurs, because macroevolution is happening on a scale that is much slower than what we can observe in a lab. So when we look at microevolution in the lab, and we can see that it's happening, we see the details of how characteristics change in populations over time. And then we look at the fossil record, in which we can view two fossils over millions of years of time. We can see the end products of microevolution. Example of this would be the evolution of the horse and the evolution of the titanotheres, the rhinos. We can see subtle changes in the fossil record, but remember, each of these changes, going from, for example, Eohippus to Oligohippus, all the way up to the modern day horse, are happening over literally millions and millions of years. So millions, thousands to millions of generations. Microevolutionary changes are occurring along this time, but we can't observe the evolution of an organism in a lab that took millions of years for it to speciate. Instead, we have the fossil record, which show us the starting point and the ending point for any particular taxa. We can also observe this in the fossil lineages of our trilobites. The various groups of trilobites, Rhydlichida, Corynexichida, Lichida, Tychoparida, and Proteids, and so on. Most of these we have a pretty good understanding of which lineage they came from. Phacopids are a little weird. But we can actually observe subtle changes in very detailed rocks, but not still on the microevolutionary level. But we can see those end members. We can see the changes that happen within Tychoparids, going from the Cambrian into the Ordovician, for example. So if macroevolution is just an extension of microevolution, so if macroevolution is the product of microscopic tiny changes that happen at the microevolutionary level, and it's just scaled up, and microevolution is driven by natural selection, we do have some legitimate questions that can pop up. For example, look at the origin of flight in birds, and what would be the competitive advantage of half a wing? And I say half a wing because according to macro and microevolutionary theory, the wing wasn't built overnight. It was an accumulation and an accretion of millions and millions of little modifications over millions of years. So at one point we would have not a full wing, but a fraction of a wing, or half a wing. So what's the competitive advantage to that? That's a valid question, but one in which we have an answer to. And that is that we need some kind of abrupt change, maybe a reproductively successful macromutation, to leap forward a little bit. And as Prothero mentions in our textbook, macroevolution is not just microevolution scaled up. There's actually moments when the evolutionary record takes a little bit of a skip or a little bit of a jump. These macromutations, so to speak. So going back to that original question, is evolution a theory or a fact? Well, it's both. Microevolution is something that's a fact. We can observe it happen. But we scale that up, not exactly scale it up, but we extrapolate that out to see the products of macroevolution in the fossil record. Okay? And not fully understanding a process does not nullify that process. Plate tectonics is a great analogy for this. We're still doing research on plate tectonics, but nobody is suggesting that it isn't happening. There are still details to work out with rates and modes of evolution. And this is where we get into philosophies of phyletic gradualism or punctuated equilibrium, which are both mechanisms in which macroevolution may occur. So the macroevolutionary process is still a theory, which, remember, a theory is nothing to be scoffed at. Okay. The fossil record provides us very clear evidence that macroevolution is occurring, but it just means we haven't directly been able to observe it because we can't sit and watch a horse evolve into a new species in real time. But it has not been disproven. And remember, in science, a theory is an explanation that is supported, but maybe can't be directly observed. And the real key here is that with macroevolution, we are using geologic time in its true context. And it helps us factor in the enormity of geologic time in and of itself. So what about the role of extinction in macroevolution versus microevolution? If we didn't have a fossil record, we would only know extinction from historical examples like moas, dodos, the thylacine passenger pigeons, and Carolina parakeets, things that have gone extinct in human history. And when we actually think about how we refer to dodos and moas and thylacines, they're commonly talked about as inferior species. In fact, even the word dinosaur is sometimes used as an analogy of something that is old, outdated, and needs to go extinct. Okay. However, opportunities by opening up niches, in other words, jobs for organisms to do following an extinction, is a key component of both micro and macroevolution. When you have organisms go extinct, it opens up an opportunity for other organisms to diversify into those ecological roles. So let's talk a little bit more about extinction, which is the main point discussed in Chapter 6. Remember that our geologic timescale is defined based on fossils and their extinctions and their appearances. Okay. We identify our eras characterized by different types of fossil faunas. Okay. So every extinction of major groups of organisms is followed by diversification and niche replacement, and extinctions mark the boundaries between these events. So for example, at the end of the Permian period, when we lost 90% of life on the planet, going into the Triassic, it was around the middle Triassic, we started seeing a huge diversification event to kind of rebound after this great dying at the end of the Permian. And that's also where we draw the boundaries for our Paleozoic and Mesozoic eras. And if we actually look just throughout the fossil record, beyond extinctions, major extinction events, we see that most species only live a few million years before they go extinct from regular causes anyway. Maybe it's just outcompeted by something else that's diversified. So I want to make it clear that we don't lose organisms only during mass extinctions. Things go extinct all the time. We call that background extinction. So why do organisms go extinct? Well, there's a couple of different possibilities. There's the basic Darwinian assumption rule that they're just unable to adapt or compete with another organism or with changes in their environment. Remember, the Earth is constantly in flux. It's constantly changing. Thus, organisms, if they're stagnant, are going to find the environment that they're a good fit in now changing into something that's not such a good fit later on. And really, there's probably lots and lots of reasons why organisms go extinct, and there's not really any that we can prove unequivocally, except for some, but again, not most of our historical examples. We can look at why certain organisms went extinct, for example, if they were hunted or captured to extinction or driven out by food. But some organisms go extinct because that plays a role, but there were other problems going on with the population and the ecosystem at the time anyway. So it comes down to, is it a matter of bad genes or bad luck? Bad genes are what we would kind of tongue-in-cheek say in regards to failure to adapt. This is organisms or populations of organisms that just can't coexist in this new type of setting as things have changed. The bad luck scenario would be affected by circumstances that are beyond the organism's population's control. This would be like taking out half of the breeding population in some kind of a large geological catastrophe or some kind of horrible earth-shattering event. That would be bad luck. Something like an asteroid impact. Bad genes would simply be it couldn't keep up with the new competition. So there's actually been some research on different modes of extinction. Jablonski in 1986 and other co-authors discussed that there are two distinct patterns of extinction that roughly kind of coincide with that dichotomy of bad genes and bad luck, except the terms they used are mass extinctions, which are geologically instantaneous. They can still take thousands to possibly a few million years to occur, versus what we call background rates of extinction. And these are just organisms that go extinct because they're not competing or able to keep up with the everyday changes that are going on in that ecosystem. So background extinctions are organisms that go extinct kind of haphazardly. Mass extinctions are a little bit less selective. Now, this work was also part of a broader series of papers. This was by Raup and Sapolsky, looking at background extinctions for approximately 2 to 4.6 marine invertebrate families per million years go extinct. Okay, so close to 5, 2 to 5 marine invertebrate families go extinct every million years. So the background extinction rate, though, declines throughout the Phanerozoic. And then in '86, the same author team found that mass extinctions appear episodically, and they suggested a possibility of a 26 million year mass extinction periodicity. But this hasn't been well received. So here's their data for our roughly 26 million year mass extinction. It doesn't really work. We see this is one of those types of situations where if you really want to see a pattern, you might be able to see one, but whether it's statistically significant or not is still, the jury's still out on that. So is there really a periodicity? Doesn't really look like one. But when we look at biodiversity of marine extinctions, so keep an eye here that for time, our most recent time is on the far left and the oldest is on the far right. And you can see that extinction intensity actually goes down. We do have major mass extinctions throughout time, but our background extinction rates do seem to go down throughout geologic history. Now, let's look at something like trilobites, for example, because these are a well-represented group of very, very diverse arthropods throughout the Paleozoic. And you'll notice that when we're looking at the gray area here's number of families, you'll notice that we see our peak diversity of trilobites around the Cambrian-Ordovician boundary. And then we see a rapid series of declines in trilobite diversity throughout the rest of the Paleozoic, going all the way to the Permian, where we have our last type of proteid trilobites still around. However, if you look at the Cambrian and Ordovician trilobites, especially you can see images of them at the bottom of the screen, you can see that they all look pretty similar for the most part. There's the weird guy agnostid on the far left that's very, very strange. But the rest of them have a lot in common. In fact, you'd have to be a bit of a trilobite expert to pick apart all those details. As diversity goes down, the disparity goes up. So diversity is the number of species. Disparity is how different those species are from one another. So we would say that in the early Paleozoic, trilobite diversity was high, but disparity was low. We had lots of species, but they all looked similar. And then as we go through the Paleozoic, we see diversity getting lower and lower, but disparity getting higher and higher. The three trilobites you see in the middle of the screen look very different from each other, yet we don't have as many species. Throughout geological history, there has been 5 somewhat episodic major mass extinction events, the 5 major events. And these are in order, not in time, but in severity. The most impactful and severe mass extinction on the planet is the Permo-Triassic. The Cretaceous-Tertiary is the second most, also known as the Cretaceous-Paleogene. This is the end of the Mesozoic era. The late Devonian is an interesting extinction because it's not one, it's a series of smaller background extinctions that were high enough that when we cluster them together, we are seeing a pretty big die-off over a short period of time. The late Ordovician is a sizable one, and then we have the late Triassic extinction, which is one of the most minor of our 5 big ones, but it's still fairly significant. There's also a couple other significant extinction events that I'd like to mention. The first is the Eocene-Oligocene extinction. This one we have a pretty good handle on because it's relatively recent, and it appears to be pretty much climate driven because the things that we see going extinct at the end of the Eocene are predominantly warm climate adapted organisms, and they're replaced by the arid grassland type organisms of the Oligocene. And also the late Pleistocene extinction, which is not one of our major mass extinctions, but it's notable because it's almost exclusive to North America and it only affects large-bodied megafauna. This is where we lose the mammoths, mastodons, saber-toothed cats, and so on. Happened very quickly, about 10 to 12,000 years ago, and we still don't have a full understanding of what caused it. It seems humans seem to have played a role in one form or another, but there's other factors such as climate playing in that one as well. But it is important to note that that one is almost exclusive to North America. So, if we don't want to go extinct, what are some characteristics that we should probably be considering? If you want to have long-term survivalship of your population, the first thing that's best to do is have a planktonic lifestyle or life cycle, preferably a larval one. And that's because planktonic organisms, or at least organisms that have a planktonic portion of their life cycle, like a larva, are widely spread throughout the planet. These are organisms that will float around in currents. So if you don't want to go extinct, don't pack all of your relatives into one place. Spread everybody around. That way, they have a better chance of finding areas where they can thrive rather than keeping everything in one place. So a planktonic lifestyle and having a widespread distribution. So being able to freely float into all sorts of areas and have a widespread distribution. Don't be too picky about climate or salinity or ocean chemistry. Be abundant. If you have a small population of organisms, they have a higher chance of going extinct. But if it's a huge population, very, very abundant, with lots of specimens and a large gene pool, you have a better chance of long-term survivorship. Living in a highly buffered environment is good too. Deep sea organisms, things that live in the ground. Being in a place where if there is a natural disaster on the surface, it's not going to affect you immediately is a good characteristic as well. And also being a generalist or a scavenger, being adaptable, not having too picky of a food source can certainly help. And I want to be clear that these characteristics don't guarantee long-term survivorship, but they will promote it. These are things that help, that we've noticed organisms that have been around for hundreds of millions of years. Most of them have all, if not most, of these characteristics. And being pre-adapted to survive stress, maybe having certain characteristics that keep the organism going in stressful environments. There are certain types of bacteria that have developed a form of antifreeze that allows them to live in frozen, sub-frozen environments like hypersaline ponds in Antarctica, and they don't die. They're pre-adapted to survive stress. So these characteristics also apply to background extinctions However, mass extinctions happen at such an extreme that sometimes it overrides characteristics in background extinction. So the characteristics we just talked about for long-term survivalhood work very well with background extinctions. Mass extinctions can be so severe that it doesn't matter. So a background extinction can, may or may not cause something to go extinct. Mass extinctions, it's a little less selective. So the best way to survive a mass extinction event, live in that highly buffered environment. Be far away from where that asteroid impact or massive volcanic eruption is occurring. And also being a generalist or a scavenger. The other characteristics are important, but they may or may not help during a mass extinction. But these two are really the big ones. So the organisms that we see that have made it through mass extinctions follow these two characteristics. They're generalists or scavengers. They have a wide, varied diet, and they live in areas that protect and insulate them or buffer them from mass extinction causes. Now, Van Valen in 1973 promoted what he called the Red Queen hypothesis, which is kind of named after a quote in Lewis Carroll's book Alice in Wonderland. Here you see it takes all the running you can do to keep in the same place. So that is the Red Queen, it's through the looking glass, excuse me. So what this essentially means is that organisms and their populations have to constantly keep changing to keep up with the constantly changing environments. So given continued and varying competitive pressures, species must constantly improve to stay in the same place, i.e. avoid extinction. Okay. So that's why even organisms that don't look like they've changed much in 500 million years, they have. It's just maybe subtle changes, but they've probably only had to change a little bit because their environment has only changed a little bit. So as the planet continues to change, life continues to change with it. And one thing that was surprising about Van Valen's research was that old species actually do have the same chance of going extinct as young ones. Just because the trilobites existed for hundreds of millions of years, they still went extinct. The horseshoe crab has been around for hundreds of millions of years. They still could go extinct if there's a mass extinction. So even our adaptable species, like cockroaches and horseshoe crabs, and lingulid brachiopods can and will go extinct at some point. So let's talk a little more about our mass extinctions. Okay. These are not coincidental background extinctions. Usually these have an underlying cause. However, it may not be a common cause. Sometimes there's multiple causes, but not always. Extinctions can be a little tricky to deal with. When you are looking at an extinction, it's important to note not just what went extinct, but what didn't go extinct. And to dive into those particulars even further, what didn't go extinct but almost did, like maybe what was affected by this extinction event, but still around, and what sailed through the extinction without any problems. So those three things, what went extinct, what didn't go extinct but almost did, and what didn't even get affected by the extinction. So what are some of our causes of mass extinctions? Now these are either things that have been hypothesized or theorized for mass extinctions or have been suggested. We have geologically instantaneous events. These would include things like a bolide or meteor or comet impact. We don't always know if it's a meteor or a comet or an asteroid, so we just say bolide, which just means big rock. So getting hit from a big thing from space is a geologically instantaneous event. Disruptions in the carbon cycle can be. So the release of solid methane clathrates can and has killed off large amounts of organisms. Ocean anoxia and changes to the thermohaline circulation system. So no oxygen in our oceans and changing how our oceans circulate are considered instantaneous because they happen at a very short time scale. Keep in mind that when we say geologically instantaneous, that can still be thousands to a million years. Or the release of hydrogen sulfides. Pretty much those toxic gases can also cause geologically instantaneous events. Cosmic Bursts or supernova, so cosmic radiation bursts or supernova, have been suggested to cause mass extinctions. And we can also look at more progressive events. So these are things that are going to cause mass extinctions, but that's because of a slow burn. So plate tectonic movements. This is going to affect sea level. It also affects climate. It affects the distribution of organisms. And significant and rapid global climate change, so changes in sea level, environmental changes, whether it's cooling or warming. This is like an ongoing thing, but eventually you get to a threshold event where we've changed the climate, we've changed the environment so much that organisms just can't adapt anymore. So let's look at some of our mass extinctions. The first up is the Permo-Triassic. This is the mass extinction between the Paleozoic and Mesozoic era. It was approximately 250 million years ago. We lost 96% of our marine species, 80% of marine life. This is pretty much, or marine genre, this is pretty much everything. We almost lost life. Things that especially were affected, crinoids, the types of corals that we saw, bryozoans and brachipod communities, were significantly affected, and they were then replaced predominantly by bivalve, gastropod, and echinoid communities. So we went from having one type of mollusk, or not mollusks in this case, but certain organisms filling certain niches being replaced by ones that were a little more effective at doing so. Trilobites, blastoids, tabulate and rugose corals ultimately go extinct at this time. However, many of those had already been in decline, so this was just kind of the straw that broke the camel's back. We then look at some other phyla that may have gone extinct at this time, such as the Tully monstrum from the Pennsylvanian, but we're not really sure what this thing is. It could be a vertebrate, it could be a gastropod, or some other kind of mollusk. So this was a huge mass extinction event that really did affect almost all life on the planet. So what caused the Permo-Triassic extinction? Well, there's lots of different hypotheses: climate change, meteorite impacts, CO2 degassing, and so on. The meteorite impact is kind of hard to test for because we don't have a lot of Permian seafloor preserved, and it would have a much better chance of hitting in the oceans than on land. However, there has been relatively recent research on the Permian and modern marine gastropods, and they showed that with massive eruptions and the low amounts of oxygen in the oceans at the time, we had changes in oxygen, carbon dioxide, and pH, and thus temperature in the oceans, and then changes in climate and ozone depletion in terrestrial settings. So this created a bit of a perfect storm to make it where organisms in the ocean had a harder time building calcium carbonate shells, and organisms on land were seeing changes in climate and in ozone concentrations. So together, this can lead to a very, very significant and massive extinction event. Up next, we have the Cretaceous-Paleogene extinction. Now, sometimes you'll see this described as the KPG. K is the symbol we use for Cretaceous and PG for Paleogene. You may also hear this one referred to as the KT. The T in that case is coming from the word tertiary, which is the term that we used to and still kind of do use for the early part of the Cenozoic. Regardless, the KPG or Cretaceous-Paleogene extinction happened about 65.5 million years ago. This is the one that we say, killed off, so-called killed off the dinosaurs. Really, to be specific, it killed off the non-avian dinosaurs, but I digress. This is actually the most well-studied extinction out of all of the mass extinction events that we have in our planet's history. A big part of that is because it's the one that killed off the dinosaurs, so to speak. But it is incredibly significant if we look beyond the dinosaurs. This is 85% of all marine species going extinct. So this is a very, very major faunal turnover. If you notice something already talking about the Permian and the Cretaceous extinction, and this is something that'll continue on with the other extinction events, we usually do gauge this in terms of how many things in the marine settings went extinct. And that's because most life on the planet is marine. So when you have some things going extinct in the terrestrial realms, that's bad. But when you have 85% of marine species going extinct, that is incredibly significant. So yes, dinosaurs are the non-avian dinosaurs, but also ammonites, rudist bivalves, lots and lots of types of marine plankton. This may not seem as dramatic as what we saw in the Permian, but marine plankton are kind of the basis of the marine food chain, so this is a huge deal. So the traditional view of this was that climates were cooling in the Cretaceous and sea levels were falling, and that's caused the reorganization of ocean circulation as we have Pangea continuing to break up. In fact, by the time you get to the late Cretaceous, the distribution of continents looks somewhat familiar to our own global distribution of continents today. That is going to change how oceans circulate, thus how we distribute temperatures and in a long-term climate. It also affects what types of ocean chemistries we have. So this is a major disruption for the marine carbonate cycle, the way that we have organisms making their calcium carbonate shells. So something very similar to what we saw at the end of the Permian. How instantaneous is this event? That kind of is up to a little bit of a debate, but we have a global iridium anomaly that was discovered by Alvarez and Son in 1979. So when you look at Cretaceous-Paleogene, what we call the boundary clay, there's an enrichment of the rare earth element iridium, suggesting some kind of an extraterrestrial source for this possibly asteroid impact. We have a crater that supports the asteroid impact. We have iridium. We have all of the things that suggest that we got hit by a huge rock from space. However, does that explain everything? Well, it doesn't. So what we can say is that the impact was an instantaneous event and clearly had a huge influence on life on the planet, but there's more to the story than just a single global killer. So we have very convincing evidence of a bolide, a large asteroid or meteorite hitting from space. And we have a big crater in the Chicxulub impact zone in the Yucatan Peninsula of Mexico. So we have lots of things happening during the Cretaceous. We have, not only do we end the Cretaceous with this big impact event, but we do have some cooling that's happening at the very end of the Cretaceous. We see continents continue to be changing in their locations, which is going to cause changes in ocean circulation, especially ocean anoxia. We see a reduction in ocean circulation, so we see a reduction in oxygen getting circulated through the oceans. We also have these big volcanic eruptions happening in what is now India and Pakistan, the Deccan flood basalts, the Deccan traps. Hundreds of thousands of cubic kilometers of basalt being erupted over about a 500,000 year time frame. This is going to have a huge influence on climate and atmospheric conditions as well. Our next extinction event is the late Devonian extinctions. Now you'll notice this one is plural. The reason for that is the Devonian extinctions are not terminal like we see with the Permian or the Cretaceous extinctions, where they're happening and actually marking the end of these geologic periods and actually geologic eras. In this case, the Devonian extinctions are a series of significant extinction events that happen to occur in the Frasnian and the Femmanian stages, the last couple of million years of the Devonian period. So instead of it being one major event, it's an ongoing trend that happens during the last few million years of the Devonian. Regardless, we're looking at a significant loss, 75% of all species, 50% of all genre. There's an important distinction there. 85% of all brachiopods. We still have brachiopods around today, but back in the Paleozoic, especially in the early parts of the Devonian, they were incredibly common and diverse. Most of the brachiopods that we lose in the Paleozoic, we lose during the Devonian extinction events. This does appear to almost exclusively affect warm water taxa, which is quite interesting. It suggests something about the cause. We're losing ammonites and most of our trilobites. Not all. We still have ammonites until the end of the Cretaceous, and they actually are quite abundant in the Cretaceous until the end. So this is the early ammonites that take a huge hit, and a lot of our trilobites take a hit here as well. But again, we're not losing all of them. Our tabulate and rugose corals, which is a specialized group of families of corals from the Paleozoic exclusively, start to kind of reduce at this time and almost go entirely extinct. Ultimately, they do a few million years after this. We see a lot of microplankton severely affected, and all of our big placoderm armored fish that are so popular to look at in the Devonian are also extinct at this point. It's not really fully clear what caused this one. There could be some geochemical causes or some geochemical evidence that suggest oceanic reorganization, could be cooling, could be impact. As we start to go back further into the geologic record, it does get a little bit more difficult to ascertain the absolute causes of these, again, because we have a reduced rock record of the Devonian compared to the Cretaceous or the Permian. Next up, we have our late Ordovician extinctions. Again, this is a long-term series of events, but it does get closer to typifying the end of the Ordovician. So kind of think of something in between the Devonian and the Cretaceous. Also affected warm water taxa, especially. We're losing 57% of our marine genre. And this does seem to coincide with a global cooling event and a global sea level fall or regression associated with glaciation, especially in the southern continents of the Ordovician period, the Ordovician world, so the Gondwanan continents. So we have this period of stable warmth and high sea levels, and at the end of the Ordovician, we drop that down. So this does have some tectonic ramifications. The uplift, for example, of the tectonic orogeny at the end of the Ordovician may have something to do with this, but this does appear to be a global phenomenon. So lots of parts moving together on here, but it does seem to have something to do with the a significant glaciation over pretty much the southern half of the planet. Finally, we get to the last of our big 5 mass extinction events, and this is the late Triassic. This is an interesting one because it's actually one of the more minor ones, which is why we have it last on the list. And what's kind of cool about this one, though, is it's happening at the end of the Triassic period. It's not enough to cause a new era, similar to like the Devonian and the Ordovician. But this happens at the end of the period that started following the biggest mass extinction event. So we have the Permo-Triassic extinction is the biggest, and then the smallest mass extinction event that still makes the list is the late Triassic, which is just a few million years later. So this almost is like a reverberation of that Permian extinction. And this is like the last death throes of it before things start picking back up in the Jurassic. Still, we lose 48% of marine genre. It's, again, another series of events rather than a single one. Also coincides with cooling and sea level fall and also increased aridity. We also have evidence from the little bit of seafloor information we have that the reorganization of the continents and the oceans during the late Triassic, as Pangaea was really starting to split apart, indicates we had some anoxic deep water conditions, and this can have huge ramifications on marine life as well. Some of the other extinction events that, again, they don't make our top 5 list, but they are significant and or interesting, and we should make note of them, is the Eocene-Oligocene extinction. Now, this is between 2 epochs in the early Cenozoic and technically the Tertiary. This is the difference between the Paleogene and the Neogene. This is that boundary. About 34 million years ago. So we're not talking about a huge biotic overturn, but it is significant and it is fairly well studied. There's a lot of events that happen at the end of the Eocene. So this isn't really at the absolute Eocene-Oligocene boundary. It's another one of these series of extinction events. What's happening here is continents are continuing to drift apart, and we're seeing climates continue to change. The Eocene is very warm conditions. For example, if you go to places in Wyoming and look at the Green River Formation in what is now arid high plains Wyoming, you're looking at tropical oil shale deposits. You find palm fronds and crocodiles and stingrays from these beautiful fossil deposits. And then when you just move up the chain to the Oligocene, stratigraphically, you're starting to see global cooling and grasslands and the drying up of all of those tropical environments. So this does suggest that we're seeing changes like cooling and oceanographic circulation alteration, which is then driving a switch going from tropical to arid grasslands. This is really when we start seeing our grasslands expand. As a result, the organisms that are living in the tropical type setting are going to be replaced by things that are more adapted to cooler, drier weather. So this is when a lot of our hoofed ungulate mammals really start diversifying as well as some of our modern bird groups. And also the extinction of our Pleistocene megafauna about 10,000 years ago. This is almost exclusive to North America. There's some weird stuff going on with megafauna in Australia as well, but that's for another time. We're not seeing a significant change in Pleistocene invertebrates. This is actually just big vertebrates, hence megafauna. We don't see a significant extinction of small mammals, so our rodents and squirrels and rabbits and things at the end of the Pleistocene are totally fine. It's just mammoths, mastodons, woolly rhinos, short-faced bear, saber-toothed cats, and dire wolves and things. All of the things that we think about when we think Ice Age mammals, those go extinct around this time. And whether it was instantaneous, because we're looking at something on a scale of 10,000 years, it's pretty quick. I mean, we don't think that the Cretaceous extinction possibly took 10,000 years. It may have been a little bit longer than that. This definitely happened at a very, very short time period. So if we look at the number of genera over time, going from about 3.5 million years ago to today, in the upper portion of this graph, you can see that the number of small mammals in North America continues going up as we go from 3.5 million years ago to today. If we look at our large mammals in North America, our megafauna, they also continue going up until we get to about 10,000 years ago, and they drop. So this is a very selective extinction. It's only affecting just this large megafauna. So we're really looking just at this point. This does coincide to around the times we're seeing an increased amount of human activity in North America, increased amounts of hunting. We're seeing the possibility of diseases being spread around, of domestication of animals. Climate is changing. So we have a lot of things that are playing into the end Pleistocene extinction and the interglacial onset of the Holocene right around this time. So what caused this Pleistocene mass extinction then? Well, there's a couple of different schools of thought on this. One is that it's caused by human interaction with these populations of megafauna, such as migrating from Asia across the Bering Land Bridge, hunting, and it also, and hunting as well. Or it could be a coinciding of that event happening with the late glacial climate change that's happening as we're reducing the amount of glacial ice and we're just changing the overall climate and setting anyway. So how might humans actually be causing this extinction? Because it might be hard to imagine that small tribes of peoples coming across, with spears are taking out huge populations of woolly rhinos and mammoths and mastodons and dire wolves and saber-toothed cats and giant beaver and camels and all of these other things. Well, anthropogenic causes are a little bit complex because you can have direct causes like hunting. Now, again, you may think, well, how are they decimating the entire population? The type of animals we're talking about that are going extinct here aren't necessarily things that you would have to take out every single member of the population to cause them to go extinct. They have very slow reproductive cycles and very slow growth cycles. Human beings, women carry a child for 9 months before giving birth. With elephants, it can be up to 2 years. Okay, and thus when that baby elephant's born, it takes a long time for them to reach sexual maturity. So if you're an early population of people and you're coming across into North America and you're hunting these big animals that can really keep your community fed and clothed and warm, and you're using everything in it, that's great. But if you take out one member of that population, if it's not sexually mature yet, there goes the next generation. So that is a thing that we do think possibly was happening, is the taking out of some of the megafauna was enough to have a huge impact down the line on their reproductive success, and thus the success of the population. We have to remember that these ecosystems, just like ecosystems today, are incredibly delicate. So we think about deforestation or poaching in East Africa today, people that poach elephants. It has downstream ramifications on the entire ecosystem of the Serengeti, for example. So yes, direct hunting can have very wide ramifications, but there's also indirect causes that could have contributed along with that. Human beings coming across the Bering Land Bridge with their populations of specific sizes, also bringing domesticated animals. We know humans were domesticating dogs and so on by this point. Those animals get interacting with the native populations, and now we start seeing the spread of diseases. And we see this happening all the time today. Invasive species having a negative impact on a broader community. So whether humans are bringing diseases or just their domesticated animals are having issues with being invasive, or the direct hunting, not to mention all of this is happening as climate is changing and we're seeing tundras turn into warmer grasslands. All of this is going to have a lot of pressure on our megafauna. and thus probably causing that extinction. So kind of the moral I want to bring about extinctions here is it's rare to have an extinction event caused by one single thing. If there's evidence for multiple causes, then most of those causes are having some kind of impact one way or the other. The hard part is teasing out which one is the main driver, if there even is one. Ultimately, when you have lots of moving parts and all of those parts start breaking down, it's going to lead to an extinction event. But how do organisms actually survive these things? How can organisms sustain in a constantly changing world? And that's what brings us to our next topic as we close out our conversation on extinctions. And now we get into functional morphology, how organisms evolve characteristics that work well in their environments, kind of summarized under the phrase form follows function. Functional morphology can be very, very complex, but we can also start it out by looking at it in its most basic terms. Really, it comes down to lessons in comparative anatomy, which was first pointed out by Georges Cuvier in 1800. You're comparing different organisms and the type of features and characteristics they have, and correlating that with types of behaviors that you're observing. Functional morphology in fossils requires a good, strong understanding of behavior and functional morphology and comparative anatomy in living or extant organisms. So we can start with what we call basically the law of correlation of parts. And while our examples we're going to provide here are very, very simple, you can imagine how quickly they become more complex. If we look at shared characteristics and their uses, for example, hoofed animals typically also have grinding teeth. So if you look at deer and horses and cows and bison today, these are all hoofed animals, and if you look at their teeth, they're teeth for grinding. So while the hooves don't necessarily have anything to do with their eating, it's a correlation of parts. Similarly, carnivores have pointed teeth and pointed claws. Now, this does usually have something to do with their diet together. So we can have correlation of parts. That if you have an animal that has sharp teeth and sharp claws, there's a good chance it was using those to eat meat. Well, how do we know? Because modern animals do that. Modern carnivores don't have a lot of grinding teeth, at least not strict carnivores. Similarly, animals that do have grinding teeth, like cows, usually don't, stalk on their prey. Now, I want to be careful here because we're also talking about homeomorphy or convergent evolution in some cases. Functional morphology can be phylogenetic, meaning it can be characteristics passed down from ancestor to descendant, but it doesn't always have to be. With form and function, you're seeing that the form the organism has follows a usable function that they are performing in their environment. For example, we have 3 aquatic vertebrates on the screen right now. We have a shark, which is a fish. We have a dolphin, which is a mammal, and we have an artist's reconstruction of a Jurassic ichthyosaur, which is an extinct marine reptile. Now, all of these are within a range that can be overlapping. They can all be a couple meters long. They are all aquatic predators. We know from a direct observation of dolphins and sharks. And with ichthyosaurs, it's based on their mouths full of very, very sharp teeth, as well as fossilized stomach contents, that these were, in fact, predators. And they have the same general body shape. If you were to ask an elementary school kid, like a first grader, to draw a shark, ask another one to draw a dolphin, ask another one to draw an ichthyosaur, the illustrations they give you are all going to be very, very similar. They're all going to have the same dorsal fin. They're all going to have pectoral fins. They're all going to have a pointed nose with big eyes. They're all going to have a Y-shaped tail. There are variances on how these features appear because they're being derived from different things. A shark is a fish. Its ancestors were fish. Sharks don't have any terrestrial ancestors that return to the water, while dolphins and ichthyosaurs do. Ichthyosaurs evolved from a terrestrial reptile. Dolphins evolved from a terrestrial mammal. So they didn't all acquire these characteristics from the same common ancestor. In fact, the common ancestor these organisms have is a fish. But if you go through their lineages, with reptiles, you have amphibian or fish to amphibian to reptile to land-dwelling organism, that one group went back to the oceans and acquired characteristics for that. If you look at dolphins, it's amphibian, reptile, now mammal, group of mammals going all the way through the Mesozoic and Cenozoic, and then going back to the oceans with acquired characteristics. So they're all taking different pathways to get to this. Now, why do they all have the same looking features, even though they're not made from the same parts? Because they're doing the same thing. This is a good shape to have if you're an aquatic predator. Okay, whether you're a fish, a mammal, or a reptile, this is a good shape to have. So the form correlates with the function. We can also observe the same types of homeomorphic or convergent evolution phenomenon in something like brachiopods. If you look at the first two on the top, Predacthornis and Dictyoclastus, these two brachiopods look very, very similar. In fact, you have to be something of a brachiopod expert or have studied brachiopods a bit to identify the differences. Same thing with Tetactronella and Caryophysis. These two brachiopods look very, very similar, but they're separated by almost 200 million years. Tetactronella and Predacthornis are Triassic, and the other two are Cretaceous. So these examples of homeomorphy, we have organisms rhyming a form. Okay, they have the same kind of structure, not because they derived it from a common ancestor that had that structure, but because both of them end up filling the same niche and doing the same job. And this is a good form that is selected by nature to work for that particular role in that particular environment. So we have to be careful when we see organisms that have different types of limbs, but they're made from the same bones. That is evidence of how evolution actually works. Okay. But when we have similar looking structures, but they're derived from different features and they have different evolutionary lineages, that's homeomorphy or convergence. And that tells us more about the function of that behavior rather than how that behavior or feature was acquired. We can see the same things with foraminifera, these single-celled protists that have been around, for hundreds of millions of years, that we see all of these different types of organisms. They're different species, but they all have very similar types of features to them, and that's because that's a structure that works well in nature, so it keeps popping up. And there's a quote that's been attributed to Mark Twain that appropriates this, is that history doesn't repeat itself, but it often rhymes. So even though they didn't derive these from a common ancestor, they keep popping up because it's a really simple, good form for these foraminifera to do the function that they're doing in their ecosystem at that time. One of the most striking examples of homeomorphy in the fossil record occurs with certain types of corals, brachiopods, and bivalves. First up here, we have rugose corals. Below that is a rictiphenid brachiopod, and over here are rudist bivalves. Now, our solitary horn coral rugose corals are exclusive to the lower portion of the Paleozoic. Our rictiphenid brachiopods are in the middle to late, especially the late portion of the Paleozoic, and our very strange-looking rudist bivalves are mostly from the Mesozoic. So why do we have three totally unrelated phyla of invertebrate organisms evolving almost the exact same shape? And that's because it works well for living in a shallow water environment. So today we have corals that are cone-shaped, that are kind of mimicking the rugose corals, even though rugose corals are all extinct. Rictiphenid brachiopods are totally unrelated to corals, and they're also totally unrelated to bivalve mollusks. However, all of these have taken up very similar forms and were also, in their own time and place, significant contributors to the formation of reefs in their ecosystems. Again, I want to differentiate what we're talking about with convergence and homeomorphy with homologous structures, or homology. Homology is when we look at parts of organisms that are distantly related or sometimes closely related, and we look at the actual internal mechanics of these individual parts, and from those we can actually see the evolutionary lineage. So in this case, we're looking at the results of shared derived characters. So we have a number of organisms on the screen. We have and they're all forelimbs. These are all arms of a seal, a pterodactyl, a bat, a bird, a saber-toothed cat, of an early Permian reptile, an ancient horse, and a plesiosaur, which is another type of large extinct marine reptile. Now, these all look quite different. However, they are all constrained because of how evolution works and how reproduction works. with the same parts. All of these organisms have a shoulder blade. They all have a humerus, a radius, an ulna, wrist bones, and finger bones. They've all just been modified throughout small microevolutionary changes over millions of years and generations to be sculpted to better adapt for a certain type of job. So for example, if you look at the limb of a seal, the limb of a plesiosaur, you'll notice that while they don't look identical, they're made of the same mechanics. They're the same parts. They have the same bones. And they have some similarities as far as a robust humerus, a robust radius and ulna, even though it's reduced in size in the plesiosaur, long, robust fingers. These are good general adaptations for swimming. So they've taken the same parts, but they've modified them into their own needs. Similarly, if you look at a pterodactyl arm, a bat arm, and a bird arm, they all are made of the same parts. They've all achieved a wing by doing something a little different, but they've all done so by elongating the fingers at some point, though birds have just fused all the fingers together into one. Pterodactyls have elongated one finger and kept the other small, and bats have elongated all of their fingers and have a webbing going in between. So this is what evolution is constrained by. It's something called a bow plan. Think of it as an architectural drawing. We have developed new designs from modifying old parts. All of these have the same bones because they got those bones from their common ancestor, but each one of these lineages has put on their own spin for a specific type of job. They've modified them a certain way, and we can see correlations between those. For example, the saber-toothed cat and the early reptile. These are terrestrial organisms that are trying to walk upright to some degree. Thus, their bones are going to be modified in a more similar way than others. So in this case, we have evidence of evolution on the fact that we're all dealing with the same general architectural plan, but it's being modified based on the role. And you can see similarities in the roles or niches or ecological jobs based on how they've actually modified those parts. So this is homology. This is acquiring characteristics over time from ancestor to descendant. While homeomorphy or convergent evolution is two organisms totally unrelated that look similar just because they're doing the same job, but they may not be modifying the same parts to do it. And these modifications can really go to some extremes, and let's look at a few examples. Here we have tarsiers, which have incredibly long, highly specialized fingers, so they can reach those fingers into holes in vegetation where there might be termites, and they can pull out the bugs and eat them. This is a very specific type of modification where they're taking the morphology that they received from ancestors, having fingers, and have modified it to an extreme. We have another example with the armadillo. The ability of a large-bodied, relatively large-bodied vertebrate to roll itself into a ball, not for locomotion, though that would be really funny, but just for protection, is really quite striking. They've specialized these armor plates and skin, something they got from their ancestors, for this ability. And if we look at some other kinds of aquatic life, it just gets crazy. The platypus. The platypus and a lot of things from Australia and New Zealand are great examples of just how far nature can push structural boundaries. Here we have, seriously, platypi are weird. We have a duck-like bill on a beaver-like body. Oh, and the males are also venomous because of a spine that they have on their back foot. Very, very... Oh, and they all lay eggs. Weird. On the right, you actually see a seahorse that looks like it's hiding inside of seaweed. That's not actually seaweed, though that's where this animal lives. This is a Sargasso seahorse. They live in the Sargasso Sea, which is the doldrums on the internal central portion of the Atlantic Ocean where we have those ocean gyres rotating. There's these very low current environments where you have lots of seaweed that just makes these huge mats in the middle of the ocean, and these animals live in them. And they've evolved this camouflage, so they've extended out part of their integument to camouflage with their surroundings to a point where they almost look like they're half plant. And then when we get into the Yeti crabs and the Dumbo elephant octopus, all of these little weird things that we never would have thought to have seen, but are apparently possible in nature because organisms over time can adapt to very, very unique types of settings. And then we get to the other side of the coin, which are organisms that have lineages that go back incredibly far, and they show very little change. Not exclusively none, but very little change. One example would be Limulus, the horseshoe crab. Horseshoe crabs have a fossil record that goes back over 350 million years. Yet, the modern horseshoe crabs you see on beaches today have changed very, very little from their counterparts back in the early portion of the Paleozoic. And the reason for that are these organisms have kind of checked the boxes on things that help them stay alive and avoid extinction. For example, they live in highly buffered areas. They have a diet of pretty much any kind of organic crap that we can find in the muds. They have a very simple reproductive cycle. So all of them, they just don't change much because they're living in an environment and doing a job that if there is change, doesn't require them to do it. They're highly adaptive. But if we think these are impressive, there's one that even goes further. Inarticulate brachiopods, the lingulids, have been around for over 540 million years. They're still alive today. They have made it through every major mass extinction and have changed very, very little. Now, you might not see a lot of these because when it comes to marine organisms, we see them usually when they end up on a dinner plate. And nobody wants to eat lingulid brachiopods because they eat all of the garbage that gets incorporated into the sediment or into the water column. So they're really kind of gross worm-like organisms that have these very simple phosphatic shells, these long squiggly pedicles that burrow down into the sediment, and they persist. They're still around 540 million years of evolutionary stasis because they're doing a job and living in an environment that does not have a lot of pressure to change even when there is some kind of environmental upheaval. They are so buffered and widespread in distribution that they can make it through. So how do some organisms do this and other ones get hyper-specialized where the slightest change to their ecosystem spells doom to that population? And this is where we get into theoretical morphology, which is something first proposed by Rupp in the 1960s. And really what theoretical morphology does is it plays around with this theoretical concept of morphospace. Morphospace is the type of fossil of variation we can see in organisms. It's the limits, or what we perceive as limits, to the shapes that organisms can have in various environments on the planet today. We plot these on another hypothetical or theoretical concept called the adaptive landscape. This is the portion of morphospace that's actually used. So think about morphospace as a thought experiment of every possible type of form an organism can have. The adaptive landscape is where we plot which of those theoretical forms actually exist on the planet Earth. Not every single morphospace is going to be ecologically possible on Earth. So it's a way of tallying up shape and form and looking at roles and function in ecosystems. So this does get into some theoretical thought experiments, but it really illuminates how different organisms, through their populations over time, take on different types of forms. So let's look at the coiling of mollusks as an example. So think of a snail shell or an ammonite or a nautilus, any type of organism that's a mollusk that has a coiled shell. Now think about the different ways you can coil a shell. That is morphospace. You can have them planispiraled, which is coiling in one plane. Think of this as if you were to take a piece of rope and coil it into a flat disc, like a coaster. This is what we see with a lot of our shelled cephalopods. The nautiloids, the ammonites, they have a spiraling that happens. Each loop is directly over the next one. So that's what we see in these cases. Trochospiraling is another form of coiling in mollusks. In this case, you're coiling over, but then there's a z-axis translation, so the coiling ends up kind of zipping off to the side. And this is what we see with gastropods and actually with some bivalves that do coil. However, in this case, we have different ways of describing the trochospiral coiling. It can be a high ratio to translation, or it can approach planispiral. So For example, our type of gastropod here, the Turritella, would be a high ratio of translation to expansion. So as there's coiling, it's kind of coiling off to the side. And then when we start getting into our cowries and even something like Neptune, we have a very degree of high to low spiraling. So there's variation just among these three on different ways that we can coil a mollusk shell. So this is just barely scratching the surface of the morphospace, the possible forms of coiled mollusk shells. So here we can look at the different types of morphospace and plot this a little bit on an adaptive landscape. So we can plot this. What type of expansion rate do we have with our shells? What's the translation degree? What's the distance from the axis of coiling? And you can see where different types of shells populate among these different parameters. Okay, so this is theoretical. The entire block diagram is theoretical morphospace. The shaded areas are the populated adaptive landscapes. So for example, we could have a shell that has an expansion rate of 10 to the 5th and a translation of 4, but we don't have anything that actually occupies that space. We could have I don't even know what that shell would look like, but you could hypothetically plot that out, but it doesn't exist. So it's theoretical morphospace. The adaptive landscape is what you actually see shaded in. So among all the possible forms, which ones do we actually see in nature? And why do we only see those and not something else? So here's another way of looking at it, but we're going to focus a little bit more on a particular group of mollusks. Let's just look at planospiral, the flat spirals, that we see in cephalopods, so our ammonites and nautiloids. What ways can we actually coil a shell in a planospiral form? Well, we have expansion here, and then we have the distance of the aperture and axis. So notice when we get down here, we have a shell that is coiled openly like this. Trying to draw on here is very difficult. Okay, and up here we have a very tight convolute single whorl. Okay. The adaptive landscape for this looks like that. Most of our shells are falling all around here. That's most of our coiled ammonite and cephalopod shells. Spirula is the weird guy that we circled here. He's down here. The spirula is a type of cuttlefish. It's a little squid-like organism that actually has an internal shell that's open coiled. It's totally different than anything else. The chambered nautilus that we all love so much is right about here. That's the chambered nautilus. Notice that sits outside of the common adaptive landscape. Most of our ammonites that we see in the fossil record fall within here, which corresponds to these shapes. Today, that's not what we see. Today, it's these two, nautilus and spirula, are the most common, and they're very, very rare because most of our cephalopods today don't have shells. So the adaptive landscape is going to change over time, and that's something else we can study. The adaptive landscape is going to change. The morphospace is pretty much going to be uniform, and it's always growing, but the adaptive landscape is always going to be changing based on the certain criteria of the environment at the time and what form is successful in those particular environments. We can discuss adaptive landscapes almost the way that we would a topographic map, even though it's not showing us anything like that. The adaptive peak is like the center of your bullseye. That's where we have the most of that particular type of form. In between, we have an adaptive valley, and that's where we see less of those forms, but that's a transition between two peaks. So these organisms are going to have a very distinct type of form, and these are going to have a distinct type of form, and these are going to be kind of in between. And that can tell us something about possibly phylogeny. Perhaps these two peaks are related. So theoretical morphology sounds pretty heavy. It's quite a lot to take in, but it really does get down, and the fossil record plays a huge role here. If we just did adaptive landscapes for what's alive today, it'd be very misleading. We have to incorporate the fossil record into theoretical morphology, because that's how we can actually understand the true nature of how different forms are used over time. Now, when we talk about form and function over time, there's two different terms we have to become very, very familiar with. The first is disparity. Disparity is the variation of fundamental shapes in an adaptive landscape. Remember, an adaptive landscape is the amorphous space that we actually observe being used. Another way of describing disparity is among organisms that have shapes, how different are their shapes? Okay, so you can say we have lots of trilobites in the early part of the Paleozoic. Yeah, but they all look the same. So while there's a lot of different kinds, their disparity is quite low. Diversity is that actual number of different taxa within a population or within a group. So we could say, again, trilobites, very, very diverse. We have lots and lots of species in the Cambrian and Ordovician, but their disparity is very low. And what we see happening later in the Paleozoic is as diversity drops, disparity goes way high. So we don't have as many types of trilobites, but the ones that we do have look really, really different from each other. So arthropods in general show this, that whether we're looking at trilobites or other things, that once they hit a maximum disparity in the early Cambrian, we haven't seen a lot of disparity since then, though it does fluctuate a bit with diversity. A lot of times diversity and disparity can be inverse. You want to see some really incredible diversity? Look at coleopterids or beetles. There are more species of beetle on the planet than anything else combined. It's really a beetle's world. However, disparity among beetles is not very high. I mean, you can look at different colors, sure, but if you look at actual different structures and forms, there's some variation, but not much. Coleopterids are all pretty similar, but they are just incredible in diversity. We can see this happening, especially with trilobites in the upper portion of the screen as we go through geologic time, so Cambrian through Ordovician, Silurian, Devonian, and then the early and late Carboniferous. The peak disparity and diversity peak diversity of our trilobites is really here in the Cambrian through the Ordovician, and after that, disparity goes up, but diversity goes down. And we see the same thing with blastoids, echinoderms, like the Pentramites and these other blastoids, that it's really in the early Carboniferous that we have their actual peak in diversity. They're around in the Silurian and they're around afterwards, but they never really hit that peak again. And again, trilobite diversity. We have our peak number of families here in the Cambrian going into the Ordovician, but this is how they all look, with the exception of the Agnostis, which is a weird little guy. The rest of these have overwhelmingly similar forms. And then as diversity starts to stepwise drop down as we go throughout the rest of the Paleozoic, we start noticing different features on different trilobites: stalked eyes, spines, robust structures on the cephalon, the ability to enroll in a ball. And then by the time we get to the very end of the Permian, we're left with this very simple type of proteid trilobite that has some of the features from around the Silurian and the Devonian, but they just don't have the extreme structures that hyper-specialize some of these from the mid-Paleozoic. So again, when diversity is high, disparity is low. And then as diversity drops, disparity starts to increase. We can look at disparity again in our shelled cephalopods to kind of go back to that concept once again. There's all sorts of different names we have for the different ways we can coil something and the way we describe the shell. If the shell is very thin, we call it orthoconic. If it's kind of fat, we'd call it orthoconic brevicone. The way that it's coiled could be ellipticonic or involute or gyroconic. These are all different possibilities. And as you can see, we actually have some really weird things with the Polytychoceras type of cephalopod. Very, very strange coiling pattern on that. It kind of goes all over the place. And that's a possibility as well, that we don't have a simple type of coil. But why do we have all those? Because of all the different types of environments, even in a marine setting, that they can inhabit. They can have spines to help anchor them into sediment. We can have more robust forms that live in the surface water interface in deep ocean environments, or the tall, skinny, flute-like shells for the open water living. Ammonites have all these different inferred habitats based on their differences in their shell morphology. So here we see one of the last of the shelled cephalopods still alive today. This is the chambered nautilus, and they're pretty reduced down in populations to pretty much the Indian Ocean, a little bit in the Pacific. They're common in aquariums and so on. You can see these things live at aquariums, but they're pretty low populations. We don't have this wide diversity of shelled cephalopods anymore. This is one of the last of that lineage. We can see the same kind of phenomenon with our bivalve mollusk shells as well. A wide variety of shell morphologies, whether it's elongate or more robust, or having spines, or growing in beds like with oysters, or in the cases of the pectins, they actually can swim. By pulling water in through their siphon and squirting it out quickly, they can flit through the water. Some of these are deep burrowing. Some of them are shallow burrowing. Some of them live at the surface attached with byssal threads. So there's all these different body plans and life positions for bivalves, again, showing the correlation between form and function. There are functional constraints, however. Pretty much why we don't see all of morphospace populated, why only the adaptive landscapes are populated themselves. And really this comes down to the bow plan, which is something we mentioned before. It's your basic body type, and it's constrained by selection and biology. If you don't have wings, for example, or if your parents didn't have wings, you're probably not going to be born with them, right? This isn't like the X-Men, where you acquire these really elaborate characteristics after just one generation of reproduction. It doesn't really happen like that. So the reason why adaptive landscapes look the way they do, why we have the populations of forms that we do, is because those are forms that are already kind of in the genome or are achievable within the genome. When we look at the morphological constraints of land-dwelling versus aquatic organisms, land-dwelling is a lot more constrained. Have you noticed that most organisms on land that are vertebrates all have 4 limbs, and most arthropods on land have 6 to 8. There are exceptions, but this is like the average, and that's because to live on land requires a lot of very selected characteristics. You have to have a skeletal structure that can hold you up. You have to be able to breathe air. You have to be able to move after your food. You have to be able to breathe. All of these characteristics have to first be achieved before we start playing around with other adaptations. So there are functional constraints, and a lot of those come down to ancestor to descendant relationships. You're not going to acquire all these characteristics just because you're in the environment. They have to be passed down with modification. So we can look at a functional hypothesis with extinct organisms are kind of hard to test. We have to be pretty careful. Okay, how do we interpret function in an extinct organism? Well, we can always do observations with modern analogs, and sometimes that's easy. If I want to know how Pleistocene dire wolves behaved, I can first start out by looking at modern day populations of gray wolves, which are very, very closely related. And then I can look for some kind of physical analog with the fossils. Okay, so physical model and model analogs in different kinds of media like air and water. For example, birds fly through the air. Penguins, a different kind of bird, flies through the water. You can think of it as flying because the physics are the same. So we can look for similar type of bone structures in fossil organisms and see if they lived in an aquatic environment and see if they may have used the same type of physics. We can also do computer models of kinetic motion, actually reconstruct some of these organisms digitally and apply some digital techniques to see how they're actually able to move. What are the limitations? This is something that's been done with the long necks of the long sauropod dinosaurs like the Brontosaurus and Brachiosaurus to see how their necks may have actually moved. And spoiler alert, it's not very much flexibility. This isn't like a snake. Their vertebrae are long, so it's more like a giraffe. They have limitations based on these types of computer models. So when we try to understand morphologic variance in fossils, it's very, very difficult because it's hard enough to do with living or extant animals. We have a bow plan, which constrains, it's constrained from evolutionary heritage or the building blocks. You're not going to get new parts out of nothing. You need new parts from old parts with modification. Ornamentation. Structures, whether they're predatory or for protective advantage, or are they non-functional ornamentation like sexual selection? By functional, I mean doing a physical function. Okay, so for example, when you look at the antlers of a deer, of a big buck, right? What are those for? Well, they're for combat with other males. Yes, that is one physical function. They are also a symbol to mates that they are fit. So they're also used for species recognition. So when other deer are walking around in the woods and they see those structures, they know what they are. So there's a wide variety with cranial ornamentation or any kind of ornamentation, whether it's feathers or hair or head structures, that can be used for sexual selection, combat, fitness, species recognition, and so on. And then pleiotropy. Pleiotropy is the idea that when you have multiple effects coming from a single gene. So there's a potential lack of a selective advantage of certain characteristics, side effects, or vestigial. These could be things like vestigial organs would be things like your appendix. It doesn't really do much, but you still have it because it doesn't get in the way of anything unless it, gets inflamed. It usually doesn't get in the way, so there's really nothing to select it out. Another example would be whales. Blue whales still have a pelvis because their ancestors had a pelvis that was attached to their spine and had limbs for walking around on land. Well, they still have a pelvis. It's inside the body, and it's just kind of floating in blubber. It's not attached to limbs or anything like that. Well, why haven't they gotten rid of it? Because it hasn't been selected out. It's not hurting anything. So there's no selection for or against. It's just there. It's just kind of hidden along for the ride. This is especially true with sexual selection and things like insects, where they will evolve a characteristic that's really good for sexual selection, but it actually can get in the way of performing other functions. So another concept here is that design of most features is not optimized. Okay, so our design of most features is not perfect. There is no perfect optimized design structure because the environment is always changing, the stage is always changing, thus there is no perfection. And that morphological structures can have multiple functions. Okay. Again, feathers on birds can be used for flight. They can be used for insulation. They can be used for sexual display and species recognition and intimidation and all sorts of things. When we want to correlate function, how an animal uses its parts, and morphology, what those parts look like, we have to be very careful. This is especially true with fossils. For example, the spirula, that coiled cephalopod that we talked about, okay, that's got the same basic coil shape, but it's an open type of coil, okay? That's an internal skeleton, which is unheard of with cephalopods. So you could look at this shell of a modern-day spirula and go, oh, well, that must have had a very, very small squid living inside of it. Actually, no, there was a semi-larger squid living outside of it. There's another group called the belemnites. It's a cephalopod that has a straight, solid shell. Well, it couldn't live in it, but the animal actually lived around it. These are from the Jurassic. So there's the eye. The spirula and the belemnite have very similar internal structures. They're some of the only ones that do. And if we didn't understand that relationship, we might make some mistakes in interpreting what these structures were used for. Now, this next part gets very touchy because in paleontology, and I'll mention it, especially with dinosaurs, this gets very subject to arm waving. Okay, how do we interpret extinct animal behavior? Really, we need two basic requirements. A phylogenetically related model is very helpful, and fossilized evidence at a high enough statistical significance that we can make some assumptions about it. And that's really tough to get. These are incredibly subject to conjecture. So we have to be very, very careful. There's a lot of things that we hear about fossilized organisms, especially when you start reading some of the popular fiction about T-Rex couldn't see you if you didn't move kind of things like in Jurassic Park, which is entirely a plot device and has absolutely no biological or paleontological evidence for it whatsoever, but it makes for a good plot device. So how can we do some of these with great care? First of all, looking at any kind of fossilized burrow, track, or trail, or a bite mark, these can be useful. So for example, these are modern photographs of organisms interacting on sandy beaches. Okay, so whether it's tracks in the sand of an organism as it was sitting down, or in this case, you're looking at tracks of a sea turtle as it came up the shore, flopped around here, laid its eggs, and then went back to the sea that way. Okay, we can observe this happening in modern animals today. We can see what kind of traces they leave behind, and we can then see how the behavior fits the trace that's left in the rock, or in some cases on other fossils. And then we can go to places where we have ancient deposits and find the exact same kind of patterns, except maybe much larger. For example, this is a turtle slide, but it's from a huge sea turtle, much larger than anything alive today. So we may not know the exact species that did this, but we can see the exact same type of pattern left behind from a very similar and known behavior. Now, what about structural capabilities to test behavior? This is one that I'm a bit sensitive to because I've done some research on this one myself, but looking at the dome-headed dinosaurs, the pachycephalosaurs. What are those big domes used for? Well, clearly for crashing into each other head-on. Well, how do we know that? Is there evidence for that? Well, we can look at structural capabilities. There was a fascinating study that came out in the around 2009, 2010, where they looked at the skulls of pachycephalosaurs and they digitized them and did what's called finite element modeling, which is structural capability testing. You're looking at how strong are the skulls, and if you hit one digitally, where's the energy dissipated to based on the properties of the skull. So it requires a lot of CT scanning and actually looking at how the bone fibers would have absorbed impact. And what they found is these things are really well adapted for that, where if you would have hit one in the head back when it was alive, it would have dissipated the energy around the skull, but not onto the brain. They also did something similar with modern known head-striking animals like bighorn sheep and Cape buffalo, and found that they do this behavior, but it actually isn't very good for their brains. Pachycephalosaurs, we don't know if they did the behavior, but if they did, it wasn't hurting their brain. So it shows they were capable of it. But I have a caveat to that. In the lower portion of the screen, you have an elephant standing on its hind legs. This is a fantastic circus trick. But elephants don't really do this in nature. Just because it can do something doesn't mean that it did it. So what would we use for pachycephalosaurs to further test this? Some kind of evidence that they were doing it. Being able to do something doesn't mean that you were doing it. Think about how many people could, run a mile in under 7 minutes, but most of us don't. So that's kind of where we have to put that separation in. It's evidence that it was capable of doing it, but that doesn't necessarily imply that that was the behavior that was occurring. Behavioral interpretations, though, usually require more than just an ability, but evidence that it's being done. Horn use in ceratopsian dinosaurs like Triceratops. These have long been suggested and used for poking holes in T-Rex. But you know what? We've yet to find a T-Rex fossilized that has clear evidence of a Triceratops hole in it. However, there is evidence of frequent injuries on ceratopsians themselves that seem to line up with horns of others. So this is a study that was done by my colleague Andy Farkey and colleagues back in 2009, where they looked at a couple of different types of ceratopsian dinosaurs, and they looked at various bones on the skulls and counted up how common injuries were that appear to be caused from horn damage, like getting scratched or stabbed, that healed. And what they found is on the big head shields of Triceratops, the side, about 1 out of every 5 or 1 out of every 6 would get poked. Out of the jugals, the cheekbones, 7 out of 39 showed injuries. None of them on the nose, and only 1 out of 45 on the back of the shield. However, if we look at other kinds of ceratopsians, we see different kinds of patterns. So they may have actually been engaging in combat and sustaining injuries at very specific parts. And again, if we look at modern head-striking horned animals today, bighorn sheep fight different than bison do, which is different than how African impala do. So we have all of these different types of evidence that implies that combat and interaction is just as diverse in the fossil record as it is today. So we have to be very careful with making grandiose assumptions about how ancient organisms behaved or interacted. It's good when you have the pathological injury evidence at high enough frequency to say something. It's great when you have evidence that the organism was capable of engaging in a certain kind of behavior. And when you get those together, you might have something to say. But we have to be very, very careful not going down the rabbit hole of too much conjecture in the ways that organisms interacted with their environments and with each other. And next week, we're going to be discussing Chapter 8, which is Paleoecology, and that's exactly what that studies. The interaction of organisms with each other and in their environments is what paleoecology is. Well, ecology is. Paleoecology is when you're doing it with fossils. Also for next week, we're going to be doing exercise 3 in lab on functional morphology. If you have any questions about any of this lecture content, please feel free to reach out for me, and I will see you in lab.